Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 3 for May 2023. Let us begin with the first question. Question 1, the photograph shows elephant grass. So this is the elephant grass they're talking about here. And they say elephant grass grows well in the nutrient-poor soils in dry areas of East Africa and is widely used as food for cattle. Its use as a source of fibers has been studied. They say the strength of plant fibers is partly due to the arrangement of cellulose microfibrils in the cell walls of these fibers. The first question says, name two types of cell found in plant fibers. Now here, because they talked about plant fibers, these are only xylem cells as well as sclerenchyma cells, so these were the best answers you could write. The other types of cells can be ignored because they are not part of plant fibers. The next question says, fibers have cell walls with secondary thickening of lignin. Describe two ways in which secondary thickening affects the properties of the cell. Secondary thickening makes the cells impermeable to water, and this is going to reduce loss of water or gain of water by the plant, and also increases the strength or hardness of the specific plant. In part B, soaking plant fibers in sodium hydroxide causes them to swell and so increases their strength. The effect of length of time of soaking on the strength of elephant grass fibers was investigated. They say the breaking strength of the fibers after soaking was determined to calculate the tensile strength in newtons per square millimeters. They say name the independent variable in this investigation. The independent variable is something that varies during the experiment. So in this case, because they're studying the effect of length of time of soaking, we can say the time taken to soak the fibers is going to be the independent variable. Next, they say, after soaking, the diameter of each fiber was measured. They want you to describe how fiber diameter could be measured accurately using a microscope. So here we need to cut specific sections of fibers as evenly as possible, and we need to cut them in transverse manner. Then we need to calibrate the microscope eyepiece reticule with a stage micrometer and use this microscope to measure the diameter of the fiber. This has to be transversely so that you can measure the diameter. Here they say give two reasons why fiber diameter was measured. Remember, this experiment is about tensile strength, and to calculate tensile strength, we need to know the cross-section area. Since tensile strength is going to be force divided by the specific area or cross-section area. So to find the area, we need to know the radius, and radius can be calculated by dividing diameter over 2. So here we will need to find the diameter of the fibers. Next, they say, some of the results of this investigation are shown in the table. Here we have the soaking time in hours and the tensile strength in newton per millimeter squared. So they say plot a suitable graph to show the effect of soaking time on tensile strength. They want you to join the points with straight lines. Because we're studying the effect of soaking time, this is going to be on the horizontal axis. So we see soaking time versus the tensile strength on the vertical axis. And the scale was quite easy to get. I just plotted the points as you see here. And this was my resulting graph. Moving on next. Here they say describe the results of this investigation. Since the question is about describe, I have to talk about the relationship we can see. Initially, we can see as the soaking time increases, the tensile strength increases, and this increase is actually linear. But after a soaking time of about 6 hours, we see there is going to be a decrease, and then it tries to flatline out after this time. So I said initially as soaking time increases, the tensile strength increases, and the increase is almost linear up to 6 hours. After 6 hours of soaking, there is a slight increase in tensile strength with soaking time until 18 hours. And after 18 hours, there is no increase in tensile strength with soaking time. So this brings us to the end of question 1. Let's continue to question 2. In question 2, scientists use pollen grains to artificially pollinate certain crop plants. Collecting pollen by hand is very time-consuming. However, bees collect large amounts of pollen in their daily activity. They say pollen traps can be used to remove pollen when the bees return to their hive. The viability of the pollen collected by these two methods was investigated, and the diagram shows a germinating pollen grain. So we can see this is going to be the stigma, and this is going to be the pollen grain germinating as the tube grows through the style. So they say give a function for each of the structures labeled A and B. We can see A is going to be the generative nucleus that is going to form the two male nuclei, and then here B is going to be the pollen tube. So I said A, the generative nucleus, it divides to form two male nuclei. Then the pollen tube releases enzymes that digest the tissues of the style in order to create a pathway to move the male nuclei towards the ovary or the ovule. Here they say the percentage germination of the pollen grains was used to determine the viability of pollen. They want you to describe a procedure to compare the percentage germination of pollen collected by the two methods used in this investigation. So since this is about pollen, I prefer to use the agar plate. So I said prepare agar and place pollen grains onto these plates using a suitable brush. You have to use pollen grains from the same species, or it should be from the same plant itself. And for pollen collected by bees, the bees should be of the same species. Then you need to add the same volume and concentration of sucrose solution to the agar plates. The sucrose solution is going to provide a source of energy and then allow the pollen grains time to germinate. This could be about 24 hours. And again, this time has to be mentioned. So it could be 24 hours, or you can choose between 1 to 4 days. During the experimentation, we have to keep the temperature, light intensity, pH, and all other variables constant. Then we use a microscope to count the number of germinated and the total pollen grains. Then using a microscope, we can count the number of germinated and the total pollen grains, and then we can calculate the percentage germination to compare the results. Moving on. In part C, the effect of temperature on percentage germination of pollen collected by both methods was investigated, and the results are shown in the graph. On the vertical axis, we have the percentage germination, and on the horizontal axis, we have temperature. We can already see that those collected by hand had a higher percentage germination at all temperatures in comparison to those collected by bees. However, each of them had an optimum temperature. For example, 30 degrees was the optimum temperature for hand-collected pollen, while here we have a little bit about 36 degrees. This was the temperature or optimum temperature for the bee-collected pollen. So they say comment on the results of this investigation. If a question asks you to comment on the results, you need to observe the data and the information and make a judgment about it. So from the data set, I wrote the percentage germination is higher for pollen collected by hand than bee collected at all temperatures. This is what I've already said. And the optimum temperature for hand collected is about 30 degrees, while that for bee collected, this should be B. Let me write B. For bee collected is about 36 degrees. Bee collected pollen had a lower range in percentage germination. You can see the difference between the highest and the lowest is quite small, while here we have the difference between the highest and the lowest is quite high. So there is a lower range than hand collected pollen. So remember, range is highest minus lowest. The reason for the decrease in percentage germination after the optimum temperature is because the enzymes are being denatured. From my observation of the results, there is no error bars, and that means no variability in the dataset. Usually, questions requiring you to comment, you should talk about error bars if they're not included, or if there is no evidence that the experiment was repeated. Here they say describe how the investigation could be modified to allow the significance of the difference between the percentage germination for bee-collected pollen and hand-collected pollen to be determined at 30 degrees. If this is at 30 degrees, that means we have to repeat the experiment at that exact temperature and ensure that all biotic and abiotic variables are kept constant. After repeating the experiment, we need to calculate the mean, and then we can observe the data to see if there is overlap in the results. And finally, we make a conclusion about the validity of the results. You could also perform specific statistical tests in order to establish significance of these results. In part D, the effect of pH on pollen tube growth of pollen collected by these two methods was investigated, and the results are shown in the graph. So in this graph, on the vertical axis, we have tube length after 2 hours in micrometer, and on the horizontal axis we have the pH. We can see that as pH increases, there is an increase in the tube length until again an optimum pH. Here we have the optimum pH 7 for hand collected, while the optimum pH is 8 for bee collected. Calculate the percentage difference in pollen tube length between the optimum pH for hand collected pollen and the optimum pH for bee collected pollen. So the pH for hand collected pollen is 7. You can see this is the optimum pH because that's when we had the highest result. You can see the highest result at pH 7, and we see the tube length is going to be 1000 micrometer, while at 8 we can see that is the corresponding tube length, which is about 750 micrometer. So the difference in tube length is going to be 1000 minus 750, which gives us 250 micrometer. So the percentage difference should be 250 divided by that times 100, or 250 divided by that times 100. So both answers are acceptable because it was just a percentage difference, not increase or decrease. And then they say suggest one reason for the difference in tube growth of pollen collected by the two methods. If you can go back to the results, you can see there was less growth for pollen collected by bees, and that could be maybe the bees released some chemical that destroyed or damaged the pollen. So this could be because the pollen was damaged, and that's why the results were the way they were, or the differences that we saw in the two results. So this brings us to the end of question 2. Let's continue to question 3. In question 3, they say scientists study the possibility of antimicrobial properties in plants, and the effect of extracts of roselle plant and clove plant on the inhibition of the growth of the bacterium E. coli was studied. The extracts were made in water and in ethanol, and the results are shown in the graph. So here we have on the vertical axis inhibition of growth, and then on the horizontal axis we have the corresponding antimicrobial agents from roselle as well as clove. We can see that overall roselle had a little bit higher inhibition effect compared to clove, but the antimicrobials extracted with ethanol had the highest effect in comparison to those extracted with water. So here the question says the method used in this study involved aseptic techniques. This explains 2 reasons why aseptic techniques were used in this study. Usually aseptic techniques are used to prevent contamination of the culture or to prevent the spread of the bacteria into the lab environment as well as to the person who is carrying out the experiment. So I say to prevent contamination of the culture by other microorganisms. This could be from the lab air or from the person operating. And then to prevent the spread of E. coli, remember in this experiment they used E. coli, from the experiment to the lab. Next they say describe a method to obtain the data in this graph. If I go back, this is the data in the graph, and of course they used water as well as ethanol for the 2 antimicrobials. So I say the results are obtained by bacterial culture with aseptic techniques. This gives you 1 mark. To do that, we prepare petri dishes with a suitable agar medium, and then we use an inoculating loop to add the E. coli onto the agar. You have to soak uniformly cut filter papers into the antimicrobial extract of each solvent for the same period of time, and then place them onto the agar plate using sterile forceps. You will incubate the petri dish at the same temperature and for the same period of time. This is for all antimicrobials you're trying to experiment on. So I use an example of 24 hours because usually they require you to stay the time. And then measure the diameter of the zone of inhibition in order to make a conclusion about your results. Here they say, draw a table to show the data in the graph. So we have inhibition of growth. There is roselle as well as clove. And when we go back to the graph, here we have the inhibition as 21.0, 15.5 if you're using the vertical scale, 17.5 and 13.0. So those are the values I put in my table for each of the solvent that was used. Then they say calculate the ratio of the inhibition of ethanol extracts or roselle and clove. So the ratio should be roselle to clove, which is 21 to 17.5, and this is going to be 1.2 to 1. So that was my answer. I divided through by the smaller one, which is 17.5 by 17.5. You get 1.2 to 1 as the ratio. Next, they say describe 2 conclusions that can be drawn from the results of this investigation. The conclusions are similar to the things I wrote here in the beginning, which are these ones here. So I said roselle extracts have better effectiveness than clove extracts, and the extracts made with ethanol give better antimicrobial effect or inhibition than the extracts that were made with water. Lastly, in another study, the effect of these extracts on the internal pH of the cells of E. coli was investigated, and the internal pH of the E. coli was reduced by each of the extracts. Suggests why these extracts have an antimicrobial effect on the bacteria. Here they say that the internal pH of the E. coli was reduced, and we know change in pH affects enzymes, so I say the pH was not optimum for growth of E. coli. This could be because proteins, which are enzymes in the E. coli, were denatured, and denaturation could be the bonds holding the 3D structure of the protein, which is the enzyme. Like ionic bonds, hydrogen bonds were broken. Once denatured, growth could not continue. So this brings us to the end of question 3 as well as to the end of this paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.